Welcome to the session 3 of discussion of structure of RNA. In this uh, session, we shall learn about RNA types, structure, and how each fulfills distinct cellular functions. If I have to look at the overview of the entire session, we shall see that RNA is a single-stranded composed of ribonucleotides. Its structure gives flexibility and function, and the ribonucleotides are joined by the phosphodiester bonds. This is a basic overview of the entire session here. Now, if I look at the components of RNA, I shall look at the components of RNA in terms of ribose sugar, phosphate groups, then adenine, uracil, guanine, cytosine, then it replaces thymine. Now, whenever a replacement has been done, it makes RNA less stable than DNA. This is a main point to be remembered, that it makes less stable than DNA. Moving ahead, if I have to see what does the RNA or how does the RNA look like, uh, basically it looks like a single stranded one which has sugar phosphate backbone. Nucleobases are present. Now, this one is a pyrimidine. The next two are purines, and the last one is again pyrimidine. If I have to compare with the rest of the first one and the last one in terms of structure, what do I look at it? I look at with nitrogen alternative positions. I have a keto group here. I also have a keto group here. The main difference comes with amino group and keto group. In case we have to see what is the difference between the first one and last one, I shall see that amino group is replaced with keto group. Wherever amino group is present, we shall use with respect to the terminology of cytosine, and the last one we can use it as uracil. Now, this is the basic idea about the structure of RNA. And what are the various types of RNA which are being present? The different types of RNA which are being present are with messenger RNA. Then I have with the transfer RNA, the ribosomal RNA, and snRNA. What do these smaller terminology stands for? The messenger RNA is the one which carries the code. The transfer RNA is the one which brings amino acids. The ribosomal RNA is the ribosome component, and snRNA is the ribosome processing of it. So these are the different types of RNA we have, and they differ based on their shape. You can visibly see how does the shape of messenger RNA, transfer RNA, and ribosomal RNA differ with. Now, what is the main part of uh, them, and how do we integrate with respect to the transfer RNA? Now, whenever we are trying to write with respect to transfer RNA, there's a lots and lots of meaning for the transfer RNA to be seen here, right? Now, whenever we discuss more about transfer RNA, let's say we have to start with what exactly are involved or how many nucleotides are involved. The transfer RNA is a small RNA chain of about 80 nucleotides that transfers a specific amino acid to a growing polypeptide chain at the ribosomal site of protein synthesis during translation. Earlier in the session or earlier session, we were discussing about translation. So during translation, at the ribosomal site, and specific amino acid is transferred to a growing polypeptide chain. That's how the transfer tRNA functions. It has sites for amino acids attachment and anticodon region for codon recognition that binds to a specific sequence on the messenger RNA. through hydrogen bonding. This is the basic idea about transfer RNA. If I have to further look at what can be the functions of ribosomal RNA and messenger RNA. The ribosomal RNA is a catalytic component of the ribosomes. The ribosomal RNA is the component of ribosome that hosts translation. The eukaryotic ribosomes that host translation, the eukaryotic ribosomes contain different uh, ribosomal RNA molecules like 18S, 5.8S, 28S, 5S. Three of the ribosomal RNAs are synthesized in the nucleolus, and the one is synthesized elsewhere in the elsewhere. In the cytoplasm, ribosomal RNA and protein combine to form a nucleoprotein called ribosome. The ribosome binds messenger RNA and carries out protein synthesis. Several ribosomes may be attached to a single messenger RNA at any time. Nearly all the RNA found in typical eukaryotic cells are ribosomal RNA. This is the basic idea about ribosomal RNA that we use the word scaffold. Whereas the messenger RNA, transfer messenger RNA or tmRNA is found in many bacteria and plastids. It tags the protein encoded by messenger RNA and lacks stop codons for degradation and prevents ribosome from stalling. Now, this is how we look with respect to RNA and RNA different types here. Now, what does messenger RNA do? This is a pictorial representation of what mRNA can do with. Now here, within the nucleus, whatever is the information is being given, that is been transferred by messenger RNA to ribosome. So what the picture representation says, the information for coding amino acids is copied by messenger RNA and it is transferred to ribosome at the location of coding of amino acids that takes place. This is a basic idea about it. Now here, whenever we are using the word with respect to folding about it, folding, hairpin loops, intramolecular bases, or lot of them. Now for you to have an idea about it, this is how the processing can be done or folding can take place. Now here, these are the different folding which have been taking place here. Now, why do we study about protein folding or RNA folding uh, uh, pathways? This is the exact one where the dynamic scope is present for research students or for data interpretation or machine learning techniques to be applied, supervised learning, non-supervised learning to be applied, and preferably a supervised learning to be applied rather than non-supervised learning. And lots of machine learning techniques can be uh, applied in order to study the folding pathways of RNA. And that's the reason we require with respect to this. Now, if I have to see with respect to RNA synthesis or RNA, uh, I'll say processing. Many RNAs are involved in modifying other RNAs. Introns are spliced out of pre-messenger RNA. Then there have been multiple spliceosomes which contain several small nuclear rRNAs or processing RNAs or the introns called ribozymes that are being spliced by themselves. RNA can also be altered by its nucleotides, modified to nucleotides other than adenine, cytine, guanine, and uracil. In eukaryotes, modification of RNA nucleotides are in general directed by small nuclear RNAs. This is how we can generalize with RNA processing here. I have a reflective question for uh, you all. What we require for the reflective questions is that why is RNA instability useful in biological systems? You can consider to answer the reflective question in terms of gene regulation and adaptability. I also have a reflective discussion to be carried out in the group discussion. The link has been given below at the end of the session. Uh, all of you all can participate in group discussion, and you have to respond to at least to two other group discussion answers or group discussion views which have been aired, and then the evaluation process will be done. Coming back to the reflective question here, discuss how RNA transient nature RNA transient nature supports cellular control and evolution. This is a reflective question here. Now you can discuss the discussion based on the guidelines which are being given. The 2' alcoholic group reactivity and the degradation which allows gene control. On basis of these two, you can always try to discuss here. And what is the conclusion for the entire discussion part of it? RNA's dynamic reactive structure enables it to act as a messenger, catalyst, and regulator. This is how we can always try to reflect about it. Now, adding a word or two to with medical applications, number of medical applications are there. The messenger RNA vaccines have emerged as an important new class of vaccines, especially what we have seen during COVID-19 pandemic. Now, apart from that, protein coding messenger RNAs have therapeutic replacement, particularly for torrential protein expression. So, some idea about the application part of it. Um, with this, most of the areas of RNA in a brief way has been considered or covered. And the references for this session have been taken from E.G. Patashala, HMI, Bio Interactive, NPTEL Biochemistry. All of them are open source or open education resources. Do participate in the quiz and do give your feedback at the end of the session. Hope you had a good interactive session. I'll be free to answer all your questions. Uh, Feel free to put them in the discussion group. Thank you.